This video will begin our discussion of Mendelian inheritance. Gregor Mendel is often considered the father of modern genetics. He lived as a priest in what is now the Czech Republic in the mid-1800s. As a priest, he had lots of free time, which he devoted to raising pea plants and documenting patterns of traits that he saw. What really set his work apart was the meticulous notes he took documenting the frequency of different traits in pea plants across a host of different experimental crosses. As foundational as his work was, it is amazing that during his lifetime it attracted no attention or acclaim and was in fact lost to science for about half a century. Hypotheses of inheritance in Mendel's time were different than they were today. The dominant one at the time was called blending inheritance, which replaced an earlier hypothesis by the great Hippocrates. Hippocrates proposed pangenesis, where each part of the body makes little seeds of itself that converge and are transmitted during conception. While this sounds ridiculous, given what we know today, for many years there wasn't a better explanation. That is, until Joseph Kohlreuter, a German botanist in the late 1700s, proposed the blending hypothesis. The basics of this hypothesis were that parental traits blended together in their offspring to form traits that are intermediate of their parents. This was the dominant inheritance hypothesis at the time of Mendel. Let's take a minute to dive into blending hypothesis and ensure we understand just what this hypothesis entailed. So, this hypothesis said that parental traits were blended in offspring. But importantly, this means that they were fundamentally changed. So, a cross between a tall and a short plant would yield medium plants. However, blending hypothesis says that the offspring does not have genes of the parents, but a completely different and changed set of genes. This medium height offspring does not have tall and short genes like its parents, but only medium genes, and can thus only yield medium offspring. And these tall and short traits are now lost. Of course, this is not what actually happens, and likely what Kohlreuter was documenting as blending were simply polymorphic traits, a topic for another video. What Mendel's actual results showed was instead particulate inheritance. This states that the particles of inheritance, which we now know to be genes on chromosomes, are passed to offspring unchanged. So an offspring does not have fundamentally different genes like was proposed in blending hypothesis, but rather a mix of genes from the two parents. Mendel's experiments confirmed this by showing that in the third generation of his crosses, he would get both parental phenotypes returning and at a predictable ratio. So before we dive into Mendel's experiments in the next video, let's take a quick look at his model organism. He chose the pea plant, which was an excellent model organism for several reasons shown here. Importantly, these characters, such as low expense, short generation time, and easily identifiable polymorphic traits, meant he was able to get phenomenal replication in his studies. In the first set of his experiments, he studied 7 characters, which each had 2 variants. By crossing plants with variants of 1 character at a time, he performed single factor or monohybrid crosses. He followed these plants through 3 generations, known as the parental generation, the F1 generation, and finally the F2 generation. Make sure you are familiar with this terminology, as it will be used throughout our study of Mendelian inheritance.